All right. Hello, everyone. It's really weird not being able to see you, but I'm going to start sharing my screen here in a second. So just give me a moment to bring up my presentation. There it is. Nope, moved away from me. Back. All right. Hi. So we've only got an hour here, so we're going to try to make this introduction part really quick before we head into breakout groups. But we are the Mixotrophs and Mixotrophy working group with OCB, subheader driving the direction of future mixotrophic research. So to start off with a quick introduction of who at least the leadership of this group is. So I am Nicole Millette. I'm an assistant professor at VIMS, obviously with a huge interest in mixotrophy and seeing where it goes.
I'm Becky Gast, and I'm a scientist at the Woods Hole Oceanographic Institution, and I have been interested in polar mixotrophy for a while. I'm really excited about this group and all the things that we're learning.
Hi, everyone. I'm Jessica Liu. I'm a research oceanographer at NOAA Geophysical Fluid Dynamics Laboratory, and I'm interested in mixotrophs as it relates to biogeochemical modeling, plankton dynamics, and just looking at effects of climate change.
Hi, everyone. I'm Holly Moeller, and I'm at the University of California, Santa Barbara. I'm interested in the evolutionary origins of mixotrophy and also how mixotrophs are going to evolve in response to changing climate. And this has been a super enjoyable working group and really stimulating research conversations to be part of, and really looking forward to hearing what all of you here have to say today as well.
Hi there, I'm Karen Stamieszkin. I'm a postdoc at the Virginia Institute of Marine Science and also an adjunct investigator at the Bigelow Laboratory for Ocean Sciences. I'm a plankton ecologist, and I'm really interested in how mixotrophs change the flow of energy through food webs, as well as the implications that they have for biogeochemical cycling.
Great. So hopefully, I assume all of you have watched the presentation before coming to this session, but on the offhand chance that maybe a couple of you didn't quite get around to it, I'll give you some of the highlights. So the important thing is how we define mixotrophs in this group, and that is that a mixotroph is an organism that gets its energy and can grow and reproduce using nutrients from phototrophy through photosynthesis, or phagotrophy through direct ingestion of prey or particulate organic matter. So the key thing that's not included here is osmotrophy, and we want to be clear that that doesn't mean that osmotrophy is not important. It's just that to make mixotrophs more separate from a lot of other phytoplankton, a mixotroph in and of itself is something that just ingests prey and can photosynthesize. A lot of mixotrophs can also do osmotrophy, but a lot of other phytoplankton can also do osmotrophy, so it's just not a requirement to be a mixotroph. And really just, I think this is a good way to sum up a lot of what the group is. Looking at a larger context is a need to understand how different groups of mixotrophs impact biogeochemical cycling and how to incorporate this into models. And so there's a lot of different ways to group mixotrophs, and when you do those groups, how is biogeochemical cycling then impacted? And we asked everyone to consider how do you think your work could interface with mixotrophs and mixotrophy? How could you consider mixotrophs in the work that you're currently doing? How would mixotrophs impact, your field of interest if it turned out that mixotrophs were a huge part of the plankton community? Things to keep in mind, and we'll get to that in the breakout groups. But actually, we're going to switch over now to a mentee meter quiz. I forgot to put the code here, but that's okay because it's going to be on the screen. So I'm going to stop and bring up a different screen now, if you will hold with me. And so for those of you who have not used a Mentimeter before, on your phone or on your computer, you can go to www.mentee.com and you'll be prompted to enter in a code, and you can see the code here up in the corner. And once you enter that in, you are able to answer the questions and the results will pop up here on the screen for everyone to see. So we've just got a couple questions here to get people started thinking about mixotrophy before we head into breakout groups and have discussions among each other. And people for this question are able to submit multiple answers. So after you submit one, you can submit another one.
It's really exciting to me to see the complexity of mixotrophy really embraced and recognized by this group.
It is definitely complicated. There's a lot of kleptoplasty too, looks like coming in, and dinoflagellates, which is always warms my heart to see.
Yeah. And we're also seeing the importance of mixotrophy to the carbon cycle. And for many of us, that might mean in two directions, both as remineralizers of organic matter and as fixers of carbon dioxide. So seeing that come up centrally, which fits with our theme of OCB.
Yes, yeah, the multiple pathways there. Someone said fun. I agree, they also are fun. And a Swiss Army knife as well, some of the smaller ones. All right, looks like it's slowing down in there. We've got a lot of answers coming in, but I'm going to move along just so that we don't spend all the time on this one question. This is great, and we'll be able to these results will be saved, and we'll be able to hopefully share them in some capacity. So this next one is there's four statements here, and you can rank each statement from 1 to 5, 5 being you strongly agree with it, and 1 being you strongly disagree with this statement. And then we'll be able to see the distribution of answers as they come in. And also, if for some reason you don't want to answer a question, you're also allowed to do that.
Thoughts on some of these questions or rationales, feel free to also drop in more comments in the chat. We know that this is not as interactive of a platform as if we were in person, but we can use the chat to also hear some of the other thoughts that you have.
Yeah, it's interesting. There's definitely a an idea that there's maybe not as enough new research on mixotrophs occurring. Maybe there could be more, but that the new research coming out, a lot of it is groundbreaking, but there probably could be better methods. And then there's at least the start of a bimodal distribution in interest of working on actual grants related to mixotrophy in the group here. So, all right, that seems to have slowed down. So we'll go on to these next questions. These are short answers. So we've only got 250 characters. So it's not meant to be very involved, but just, you know, if you think about it, first thing that comes to mind, what do you think should be like immediate research goals for mixotrophy? So stuff that should be happening right now and where focus should be at. Oh, a lot of interest in biogeochemical modeling. All right, a lot of great ideas coming in right away.
I think this also highlights what a lot of the open questions in the field are, like the basic questions of where mixotrophs are found and the magnitude of their importance.
A lot of interest in incorporating mixotrophs in models, a lot of new methods I'm seeing, looking at where mixotrophs are having impacts, characterizing photosynthesis biomass versus prey biomass. A lot of stuff for in situ detection of mixotrophs, studying them in the environment. Who is doing what? Yep. All right. People can feel free to potentially still fill. I think you might be able to still fill some or add something to that question before you move on if you were still finishing typing up something. But the next thought is what should be long-term goals for mixotrophy? So this would be stuff that maybe we don't even have the capacity to do this right now, but you know, just thinking way in the future, what should we be working towards? Much bigger picture understandings of mixotrophy or how they're studied.
It's really neat to see the intersection of these long-term goals with some of the shorter-term needs in the field and how, for example, developing methods for in situ characterization could help us with a more robust understanding and figuring out what some of those gene markers are.
Mixotrophs from space. Questions related to climate change, yep. Definitely see, yeah, a lot of focus on taking like a lot of what we learn and applying that to predictions for how mixotrophs are impacted or impacting long-term changes related to human impacts. Okay, you seem to close. So we've got one last question. It's another word cloud. And so we've thought about some of these goals, short-term and long-term. And then what kind of tools do we need to meet these goals? All right, everyone's about omics these days. I can't say I disagree. In situ rate measurements, I definitely second that as well.
And those can definitely be folded into the mechanistic models that then can loop into some of the shorter term research goals of incorporating mixotrophs into biogeochemical models.
Also, sorry some of these colors are not showing up as well in this background. We just picked a theme and didn't quite properly test out some of the colors.
And there's definitely two streams of approaches to, right? The culture-based methods, can we diversify what we can bring into culture and work with in the lab? And then also a need for better field tools to go out and measure rates and fluxes in situ. It's neat to see those two themes emerging, and it's really consistent with a lot of the discussions we've had as part of the working group.
All right then. My screen, which I have now. Okay. Great. So thank you guys for taking the time to do that. We're going to move into breakout groups now. So for people who have been attending a lot of these sessions over the past 2 weeks so far, it'll be very similar. So find a table. You know, if you're just at a table with a couple people, maybe try to join up with another table, and you can open up the whiteboard, and there will be three questions in there, and then you can below there will be tiles where you can enter in your response to each of those 3 questions. There's also a place where you can enter in the name of each person in the group, and also we will go into breakout groups for about 30 minutes, and then we will come back for a quick just report from each one. So if you could quickly elect a reporter for the group, and then put into the chat the table, the floor and table, and who the reporter for that will be, so we know who to call up on stage to give that report. Is there anything else you guys think I have missed? All right, and we will also find our way to a table with an open spot to join in the discussions as well. So we're looking forward to talking with people. So I think that's it, and we'll be ready to go so we don't waste any more time. We'll move to the breakout groups now.